Good day, this is Professor Will, CBMD PhD. Today's Tuesday, September 29, 2020. It is 8:53 and 27 seconds p.m. Eastern Standard Time. It is a continuation of my microbiological research, particularly phylogeny and prokaryotes. Let's hop to it here. We will go about this in a top-down fashion, starting with the major eubacterial and archaeal taxa, leaving the details and definitions of the lower subordinate taxa to you and the next generation of microbiologists. I do not claim that these high-level taxa are currently defined are entirely correct, are genuine naturally defined groupings, in all cases, but most of them surely are, and therefore, our discussing them at this point is useful. Let us start with, as we have already seen, the archaea represent a unique cell design. In their cellular organization, they stand apart from the eubacteria, and of course the eukarya. The archaea are also unique in their overall phylogenetic structure. The domain comprises two characterized kingdoms, the Euryarchaea, the Crinaarchaea, with the possibility of a third kingdom, the Characheida, lurking among the unwashed masses of a species that have been identified only through direct isolation of tRNA genes from the environment. Even a fourth archaeal kingdom is suggested by the recent discovery of a bizarre nano-sized exosymbiont associated with particular ones of the Crinaarchaea. What makes the archaeal phylogeny stand apart from the eubacteria and the eukarya is pronounced phenotypic and genotypic distinction between the kingdoms Euryarchaea and Crinaarchaea, the depth of their phylogenetic split, the genomic difference between them. Inter-kingdom differences of this magnitude are not seen in the other two domains. The Euryarchaea appear phenotypically constituted designed around a methanogenic metabolism. They are also characteristically thermophilic and show relatively high intercellular concentrations of monovalent ions. To our present knowledge, the kingdom has spawned four main methanogenic branches: phylae, the methanococcal, the methanobacterials, the methanomicrobials, and the methanococci. The Euryarchaea kingdom also harbors other non-methanogenic phenotypes. Interestingly, all but one of these offshoots of the lineage that gives rise to the methanomicrobials. The non-methanogenic phenotypes in question are the extreme halophiles, the sulfite-reducing archaea, archaeoglobals, and the thermoplasmatels. The lone non-methanogenic group not stemming from the lineage is the Thermococcus, which comprises the genera Thermococcus and Pyrococcus. The extreme halophiles are facultative aerobes that grow in highly saline environments and have been known to microbiologists for ages. They are known to putrefying salted fish, among other things. The extreme halophiles are also notable for the reddish pigments they produce, which are in part responsible for the striking red color in salt evaporation ponds. Among these pigments is bacterial, now archaeal, rhodopsin, which provides them a photochemistry. Archaeal rhodopsin is essential to light-driven transmembrane, pumped by means of which the extreme halophiles can derive energy. Beyond their extremely high 5M intercellular potassium ion levels and their photochemical ability, unique among cultured bacteria, the extreme halophiles offer little of interest, unless it be the fact that, as an organismal group, they are phylogenetically very tightly clustered, and so of relatively recent origin. One naturally assumes, given a specific though somewhat distant phylogenetic relationship to the methanomicrobials, that the metabolism of the halophiles is probably a derived degenerate form of the metabolism characteristic of the methanomicrobials. But the halophile genome shows something different. It is rife with bacterial genes and others that have no relationships functionally or phylogenetically to methanogens or methanogenesis. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.